Thank you. So I'm going to talk about a project that we're doing for sickle cell disease. And just a little background, sickle cell disease affects about 100,000 people in the US, disproportionately affecting minorities. And medically, it causes severe medical complications, including pain crises, acute chest syndrome, splenic infarcts, renal failure, and stroke. So despite advances in medical care over the last decades, sickle cell patients in the US and in California in particular still leads to early mortality with an average lifespan remaining about 40 years over the past decades. So this is the pathophysiology of sickle cell all in one slide. So red cells are bags of very high concentrations of hemoglobin, which is typically an alpha-2 beta-2 tetramer. And in sickle cell, every patient has the same mutation, the 6th amino acid. It's changed from a hydrophilic glutamic acid to a hydrophobic valine. And so at the upper right, what you see is that valine winds up being on the surface of the deoxyhemoglobin tetramer and can connect with an adjacent hemoglobin through a hydrophobic pocket between phenylalanine and leucine 8885. And that then leads to polymerization, aggregation, and comes out a solution. The red cell turns from being a flexible a spheroid to a sickle stiff cell that then causes vaso-occlusion. So the approach that we're taking is based on observations about the effects of fetal hemoglobin. So as you probably know, the adult beta-globin is one gene from a whole complex that's expressed sort of from left to right through ontogeny, with the epsilon being expressed in embryonic gammas during fetal and the beta in adult life. So that adult hemoglobin is beta-2, alpha-2 tetramer. But in fact, we all express a little bit of fetal globin, so that most of our hemoglobin is A or beta-2 alpha-2, but we all have a variable amount of hemoglobin F, which is epigenetically determined. And in fact, an observation goes back 20 years that patients who express more fetal hemoglobin, in this study they used a cutoff of 8.6%, actually lived a decade longer. And the explanation is shown on the right. So where there's that hydrophobic pocket on the adjacent hemoglobin tetramer, fetal globin has this hydrophilic glutamine there that prevents this axial contact. So in fact, fetal globin is dominantly anti-sickling. And so we're going to express a beta-globin that has that glutamine 87 and other amino acid changes to make it have the anti-sickling activity of fetal globin. And so what we're talking about is a combined product that's both a gene therapy and a stem cell therapy. And the idea is to introduce this anti-sickling beta-globin gene into the hematopoietic stem cells of an autologous from patients with sickle cell disease, so that when transplanted, they'll now have a permanent source of stem cells that will make normal erythrocytes. And the vector that we're using is shown here. So it's a lentiviral vector that has this cassette of the human beta-globin gene, including its locus control region elements. And it has 3 amino acid substitutions that give it potent anti-sickling activity. So it has that glutamine at amino acid 87 and 2 others that have been shown in other hemoglobins to prevent sickling. And so this AS3 or anti-sickling 3 globin produces this anti-sickling version of beta-globin. And because the control elements are from the globin gene, it's expressed only in erythroid cells, even though it's present in all the hematopoietic lineages that come from the modified stem cell. And so the way the cell processing is done is bone marrow is collected from the patient, the hematopoietic cells are enriched for CD34, and then cultured with a combination of recombinant cytokines, in this case c-KIT ligand, FLT3 ligand, and thrombopoietin, that activates the stem cells so they're susceptible to taking up the lentiviral vector. And it's just a 2-day culture period. So the project that we've done was funded by CIRM, and this is sort of where we started about 4.5 years ago. We proposed in a disease team 1 proposal to take the parent of that vector that had been only tested in mice and evaluate over the course of 4 years whether this would show efficacy in human cells from patients with sickle cell and to bring to the point of IND. And so our timeline is shown at the bottom with pre-IND after the first year, getting to an IND in 4 years. And we made it with just 3 months over this timeline. And some of the preclinical data that shows the activity is shown here. So the model we established in the laboratory starts with bone marrow CD34 fractions from either healthy donor or sickle donors. We introduce the vector, and normally at the end of 2 days, that would be the patient product. But in this model, we then do an in vitro differentiation from the stem cells, so at the end of 2 or 3 weeks, we have a population essentially of pure erythrocytes. And that's shown on the bottom right that over, in this case, 87% of the cells are enucleated, not staining with the nucleotide DRAC5, staining with glycophorin A. And we can characterize expression by the vector and the effects of sickling. And so just to summarize the preclinical efficacy studies, we optimized lentiviral transduction of the sickle bone marrow cells to obtain the desired vector copy number, which is between 0.3 and 3 copies per cell, and the percent transduced at least 10%. We showed that we get a sufficient level of this AS3 expression, which was about 18% of the total beta-like chains were coming from a single copy of the vector. And we also obtained this level transplanting the cells into immune-deficient mice. And then in vivo analysis of the beta-globin gene transfer showed consistent beta-globin expression. And I didn't show it here, but we've evaluated morphologic correction. And in fact, it just seems that every red cell that comes from a modified stem cell is resistant to sickling. So we have potent anti-sickling activity in the red cells coming from the modified stem cells. We also performed multiple safety studies of this. One of the concerns with integrating vectors is insertional mutagenesis. It's not clear if a vector using lineage-specific elements would have that risk. And so we did an in vitro insertional mutagenesis assay using mouse bone marrow stem cells and showed that this vector did not cause any clonal outgrowth. The positive control, a gamma retroviral vector with a strong non-specific control element, did cause clonal outgrowth. We looked at vector integration sites in the human cells and grafted in NSG mice, and there were no insertions found near the canonical insertional oncogenesis genes that have been seen in some retroviral trials, and there was no selection for clones with integration near cancer-associated genes. And then finally, we did a formal GLP in vivo toxicology study, and we showed that with this lentiviral vector, there was a lower frequency of integration near cancer genes than the positive control, and that that with the positive control, there were clustered integration sites, but there weren't with the vector. And these results were all published last year in the Journal of Clinical Investigation. And so the idea for the trial, the schema is shown here. This would be, again, an autologous bone marrow transplant, so the patients would undergo marrow harvest in the typical way. Bone marrow CD34 cells would be isolated, cultured with the vector, and then the cell product would be frozen and completely characterized for release criteria. And if it meets release criteria, then the patients would be admitted, would undergo chemotherapy to make space in their marrow. The cells would be transplanted back into the patients. The cell processing is sort of diagrammed here. So starting with from a half to a liter of bone marrow, we plan to use the Miltenyi prodigy for initial red cell depletion, followed by the Miltenyi Clinimax for CD34 selection. The cells are then cultured overnight with a combination of cytokines to activate them, and then cultured for a second day with the lentiviral vector, with in process and final testing done, and then the cells are cryopreserved. This is data from one clinical scale run using normal healthy donor marrow, and showing that the copy numbers of the vector looking short-term in vitro in either erythroid or myeloid conditions, in colony-form units, or in primary or secondary mice is preserved, indicating that we're getting gene transfer to engrafting stem cells based on this model. And the trial that we plan then is a prospective, non-controlled, non-randomized phase 1 trial to assess safety primarily, but with secondary endpoints for efficacy of the cell therapy in patients with sickle cell disease, which would again use autologous bone marrow cells transduced ex vivo to express its anti-sickling gene. The initial trial that we plan is relatively small with an N of 6. We are going to start with adult patients with sickle cell, either SS or S beta thal, which are the most severe forms. And we would rule out anyone who has a matched sibling donor or a 10 of 10 allele donor at this time. Then we have a whole list of very specific disease severity criteria and organ function criteria, kind of typical for bone marrow transplant. And again, the primary objective will be safety of this procedure for patients who are both getting these gene-modified cells as well as receiving myeloablative conditioning with busulfan. And then secondary endpoints are for efficacy, to look at the transduction and engraftment of the stem cells, the expression of the beta-AS3 gene in red cells in circulation. We were working with someone at USC, Herb Meiselman, who could measure the red cell properties in terms of their rheology and their physical properties after expressing this globin gene. And then we have clinical endpoints to look for the standard sickle cell disease endpoints of hemoglobin, hemolysis, and clinical symptoms. So the current status of the project is shown here. Again, the CIRM funding for the first trial began in March of 2010. We then went through the whole gamut of NIH RAC, IRB, Scientific Protocol Review Committee, Biosafety, Data Safety Monitoring Board. We submitted an IND to FDA in May, and that opened in June. And then a second CIRM grant began July 1 for the actual trial performance. The clinical grade vector has been produced at Indiana University's vector production facility, and it's undergoing release testing now. And I will stop there. Thank you.